All right, back again. Part two of Europe and its Australo-Melanesian ancestry. I think part one was pretty well done. I think I was very pleased with the way that one came out. And I look to and hope to, you know, um, make part two as eventful. So part two is going to be, it's going to, partially a summary. Gonna have new information here as well, and pretty much just pick up where I left off at. Now that I've read the genome study that I built this, series off of, built the foundation for the series off of. I'm gonna go into part two now, and yeah, let's get into it, man. I'm gonna put the objectives for part two on the screen. And yeah, let me get to work. Let's go. So this is what we got here for part two. I'm going to share morphological findings from the population that we reviewed, the populations, I should say, because, there were multiple populations within the mosaic, like the genome study showed. And, just in case, people want to play the ambiguous game as, of, yeah, they might have had, you know, the C haplogroup, but that doesn't mean that they were Negroes in type. This could have happened, that could have happened. Y'all know how they get down, you know. So, in addition to that, I'm going to share an article about the early presence of Australo-Melanesians in Australia 65,000 years ago, because I said that I was going to do that in part one, and I want to make good on my word. Um, and I'm not going to read the whole article, you know what I mean, because I don't think it's pertinent for me to read the whole article, because the headlining title, says everything that we need to know about it. Then review results and conclusions of paleogenomics of Upper Paleolithic to Neolithic European hunter-gatherers. That'll give me the opportunity to share, my AI, the flow chart that I had AI design. I think is pretty nifty, and it's derived directly from the genomic study, just to give the audience a visual and breakdown, because they mentioned a lot of populations. So I figured I'd simplify it and use a visual as a review instead of just redundantly reading something. You know what I mean? I try to mix it up. I try to be, a good presenter, you know what I mean? And mix up different styles of presenting information so that it doesn't become boring, you know what I mean? So anyway, I'm going to read 2025 article on pigmentation in ancient Europe. That one I'm gonna read the whole thing, because it's pertinent to read that, because it pretty much lines up with the 2023 genomic study, and it matches up with it pretty well to show that this information is building on top of it, of each other. You know what I mean? These are not just one-offs. One particular genomic study is saying one thing that's not accepted by other genomicists. You know what I mean? So I want to share that, and I'm going to share that. I'm gonna align pigmentation timing with the population divergence timing. I'm gonna shoot that off the top of my head. I think after I get done reading the pigmentation article, I think for the most part, you all will get that gist. Explain how modern Europeans diverged. Yeah, I'm gonna shoot that off the top of my head too. I might pull the PCA chart, man. You know what I'm saying? I might do that separately. I don't want to get too far behind on schedule and putting out this part two. So that I might just do separately in a short clip, or I might be able to find it right now, time permitting. Um, How early was the Y-DNA in the Levant? I'm gonna go into detail about that with ChatGPT. That was really interesting. That thread that I started with ChatGPT. Um, but you're gonna see that the early Levant was occupied by um, what's the word for them? Near Eastern hunter gatherers is what that article called these particular people. And although they didn't identify a haplogroup with them in that study, now mind you too, when we're pulling these studies off online, these are not even the complete studies. They're not sharing the complete study in full detail online. It's very difficult to get the complete studies. If you notice in the genomic study that I had, in part one, it said it was about 120 pages, but I only had access to 9 of them. You know what I'm saying? So it's probably a lot more information in the complete study, but I think, it's enough information in the short version to get the gist of what was going on in Paleolithic Europe. All right, next point is I'm align the Levantine and West African population with the Terrafault and Natufian populations and explain how they contributed Eurasian gene flow into West Africa and thus spreading Neanderthal and the Denisovanan variants into West Africa. I'm connect this with a genome study on the depopulation of history in Africa. I've already shared that in the first part, but maybe I might share it again just to make sure I hammer that home. Why does this put pressure on the outer Africa theory? We'll talk about that. Why the European 20% ancestry from ancient Eurasian and mixing America? Now, I might just cut it off at how early was the Y-DNA in the Levant, and I might just come back and do a part three and finish the rest of it, time permitting. So I'm gonna try to keep this to 2 hours, possibly less, and we'll go ahead and kick this off, man. Let's go. So first, I'm gonna get that Australian article on the screen. I'm gonna drop this off, this PowerPoint here. Through that, and I'm gonna add something else. And I just want to share the title. Let me make sure I got it on the screen. There we go. So let me see if I can zoom it in a little bit. Actually, I think that might be zoomed good enough. Let's see. It's even better. So you can clearly see here what the article was called. Let me see when this came out, when this was published. Don't see it. It might be at the end. Let's see. Okay. 2017. All right, great. So this article is titled Human Occupation of Northern Australia by 65,000 years ago. So like I was saying before, the CF superclade, which the Melanesian lineage split from, according to AI and genomic studies, the most accepted model for the splitting is in Southeast Asia at 65,000 years. And as you can see here, Melanesians were already in Australia. Austro-Melanesians were already in Australia at around 65,000 years already. So Once again, that's putting pressure on the outer Africa theory. I personally believe that, you know, human beings didn't arise just off of, a couple people. I think that they moved in bands. I think they uh, started in smaller bands, and basically solidified certain genetics. And, they have foundational genetics, and, those genetics further mixed amongst themselves. And you eventually get more and more diversity and an effective population size over time. You know what I mean? And due to cataclysmic events, so forth and so on, which was described in this first genomic article, you get more derived populations later on in time. But I ain't gonna get too far into that. I'm gonna get to what we're getting to, and let's see what's next on the menu here. So I did the Australian article. Now we're going to review the conclusions of the paleogenomics of Upper Paleolithic to Neolithic European hunter-gatherers. So I'm gonna get the PowerPoint back on the screen, and I'm gonna break this down. It's really pretty much self-explanatory. Really self-explanatory. So I'm gonna do that. Share the PowerPoint back up on the screen. All right. I think this is a very good visual. Populations of Europe and West Asia, timeline, genetic relationships. Hold on, let me make sure I didn't forget those. Let me see here. Share article review results in 2025. Yeah. I was actually supposed to share the morphology within ChatGPT first. Yeah, I don't want to miss out on that, man. I'm out of chronological order. It's not even on the screen anyway, but let me pull this out of queue, and let me get my ChatGPT back on the screen. There it is. Let me get my thread on the screen. Okay. I just need to share that on the screen. And there we go. I'm gonna go through this. I'm gonna run through this. Like I said, this is very necessary because we have those who will definitely try to ambiguate the information by trying to poke holes and could be would be's and everything like that. Just a lot of dishonest people, man. So we got to nip that. Got to go ahead and nip that right now with ChatGPT. Using ChatGPT as with to assist me in these receipts. So in part one, we went over the craniomorphology portion of this, and let's see if I can get it. Yeah, so they were all long-headed. Okay, but just in case somebody would say, well, any individual, any population can be long-headed, in modern times, that doesn't mean, just being long-headed doesn't, demonstrate the full description of their morphology. So I went further. I had ChatGPT go further in terms of morphological index and what the facial form and the nasal apertures look like. Um, and let's see further. It's one particular thing I wanted to point out about this nasal structure. Let's see if it's on here. Yes, it's right there in front of my face. So, in my opinion, nasal structure is something that is much more non-subjective than any other, morphological category. Because to this day, no other populations have broad nasal apertures with low nasal bridges. Um, Everything else, they can probably, ambiguate, the other categories. But if you look around, people, if you look in the mirror, you know what I mean? This is not racist, this is just the scientific truth. Negroid populations, still to this day, are in a category of their own in terms of nasal structure. And these populations have broad nasal apertures and relatively low nasal bridges compared with many Holocene Europeans, and especially modern Europeans. See the graph here? Earliest Upper Paleolithic Europeans, generally broad to intermediate nasal apertures. Facial form was broad, robust faces. In the Mesolithic Western hunter-gatherer, this is when you start seeing a transition. This would have been in Cheddarman's population group. They're more of an intermediate in nasal apertures, robust faces. The early ANF, the ANF is the Anatolian farmers, generally narrower than many West hunter-gatherers, more gracile. That means their faces were less robust. Uh, Many later Holocene Europeans often narrow on average, more gracile overall. So you could see how the morphology is changing. It went from a robust facial look and nasal, wide nasal apertures, to in the Mesolithic period, you're seeing the intermediate changes and the changeover. Um, Faces are still robust, but as they mix in, and when I say they, I'm talking about contemporary Europeans, in particular contemporary white Europeans. Their ancestry is derived from Mesolithic Western hunter-gatherers, which that population, according to genome studies, was replaced by Eastern hunter-gatherers and early Anatolian farmers. These same Anatolians would also contribute the later layer of the Levant, as well. That the ANF group is where you get that you know that that modern Mediterranean um, skin tone from. I think what they call olive today. You know what I'm saying? It's kind of like it's not the Northern European extreme pale pigmentation. It's more of a man. It's hard to explain, but I think you guys know what I'm talking about. The current inhabitants of the Mediterranean today, have that skin tone, which is a little bit different than, the Northern European extreme pale uh, skin pigmentation. The Anatolians brought that skin pigmentation into the Levant. So um, there's a factor, uh, I don't know. Well, actually, yeah, the article actually pointed out that the Saikino um, introduced the paler pigmentation, which we recognize as the Northern European pale pigmentation, the palest. That's a word. Uh, Depigmentation allele. And I'm gonna ask, because that article um, determined that came. It seemed, well. It inferred that was introduced from the Saikino population. And I'm gonna ask ChatGPT: is the Saikino, might not even be spelling that correctly, but it'll understand what I'm saying, population group responsible for the second depigmentation allele, according to the genomic study. Let's see what it says. Ooh, I'm on point with that. Cool. Wait. Yes, with an important qualification that's broadly consistent with what the study in previous ancient DNA work suggests. But the study does not say that the Sidel-Kino cluster invented or exclusively carried the allele. Here's the pattern. First major depigmentation allele, the first major light skin allele to become common in Europe is generally the SLC2-4A5. The 2023 study shows that this allele increased after the last glacial maximum, just like I said, and was widespread among populations carrying Villebranca-related ancestry and later became nearly fixed in Anatolian Neolithic farmers, just like I said. Second major depigmentation allele is the SLC4582. This allele remained uncommon for much longer. Current ancient DNA indicates that it was rare or absent in most early Upper Paleolithic Europeans. Remained relatively uncommon among early Villebranca and Western hunter-gatherer populations, and increased substantially in frequency later, particularly amongst Eastern European hunter-gatherers and populations with Eastern hunter-gatherer derived ancestry. So that's the third layer. And so it's pinpointing, and it's being precise and saying it's not just the Cytokino. Obviously, the Cytokino is is coming from this Eastern hunter-gathering population. Cytokino cluster in 2023 Nature paper represents the ancestry that later gives rise to the classic Eastern hunter-gatherers. So yeah, I find it interesting, man, that adversaries, and not just me, man, they keep claiming that we don't understand these genomic studies, man. But as you can see here, man, I'm spitting this off the top of my head, and it's aligning with everything that's in that genomic study. You know what I mean? So people can claim, we don't know what we're talking about. Just like Sean P. What was interesting in what he was saying is that he this is his own words. The white man said it, and therefore it is law. Basically, he didn't say it in those exact words, but that that was the gist of it. And you guys can go back and watch part one when he says pretty much just that. And then he makes a claim talking about I'm not a genomicist, and so he doesn't have to listen to what I'm saying. Well, I'm not a genomicist, but I have a very good understanding of the basics of genomics, of genetics. enough to point out, these glaring facts in these genomic reports that's not widely spread, in my opinion, in mainstream, academia. So yeah, they pretty much just trace the ancestry makeup of what you would call contemporary Europeans today. And this is, coming strictly from genomic studies. You know what I mean? So I know people will try to again, throw that racist card in, you know what I mean? But science is science, man. Science is used against, so-called black people every day, all day. And then when you actually tell the truth about another population group strictly by the science and consistently with the science, we're racist, but people can say whatever they want to say about black folks, talking about they only come from Africa, despite the fact that we were all over the world. Our presence was all over the world, um, you know, even after the last glacial maximum. But man, the hypocrisy is real. But we gonna move on, man. Let's next here, man. ChatGPT actually answered my next question in terms of where the cytokino, what type of population they belong to. I suspected that it was coming from, the Eastern hunter gatherers, but like I said, that's it in a nutshell in terms of contemporary European ancestry. And so, contemporary European ancestry needed those three layers to derive from. You know what I'm saying? So, those who derive from the Northern European look, so to speak, I'm not being funny. You know what I mean? And I ain't even going to keep repeating that I'm not being racist, man, because the science speaks for itself once again. You are derived from three different population groups. So when we say that there was no, contemporary white looking Europeans around, at least in mass numbers, we have genomic evidence to support that. Just like we have the genomic evidence to support the presence of Negroes all over the world, post LGM and after, we have the genomic information to support that. You know what I mean? And y'all shouldn't have no problem with that, man, because y'all love DNA, like y'all say, until it betrays y'all, until the science betrays y'all. This is what the science says. And here's another summary. And I'm gonna give you another summary on top of that. So anyway, man, let me take this off the screen, and let me look at my PowerPoint and see what else is supposed to come up here. Share that Astralo-Melanesian article, conclusions of the paleogenomics. So yeah, this is supposed to be the PowerPoint portion, and I'm gonna get that back on the screen and give you guys a whole rundown of how the gene flow was coming out of Northern Europe after and during the last glacial maximum, and what that population looked like before the last glacial maximum. Screen, get this on the screen here. Yeah, let's have some fun with this, man. Double check one more time, make sure it's on the screen, and it was not. Man, I'm glad I checked this stuff, man. Yeah, let's zoom that in. Okay. Populations of Europe and West Asia. Timeline, genetic relationships, pigmentation, and lactose tolerance. Okay. So it's saying here that The deep West Eurasian ancestry um, split into two groups: the foreknown cluster and the Westernese cluster. And the gene flow, according to this chart, and obviously according to the genomic study, um, suggests that the gene flow only continue, or at least mainly continue, through the foreknown cluster, through the Solutrean associated populations, uh, to the Magdalenian and Elmoran cluster, and to the Villabrana. Now, before the Villabrana, you can see here that the Y-DNA is consistently C1. C1 across the board. And this is associated with the Gravidian culture first, Solutrean, then the Magdalenian, and then the Upper Paleolithic Natufian antecedents, pre-farming. Okay. And that's going to be relevant for later, because the Natufians, they played an important role, in terms of gene flow into the Levant, like I said. And I got another flow chart strictly for the Levant, but I'm gonna make sure I stick to schedule. I'm gonna check the objectives, and I'm gonna make sure I stick to schedule. But anyway, you can see here that the gene flow changes, makes a sharp change in the Mesolithic era, right? And it becomes I2 with the Apracaso population in the West, hunter-gatherers. Okay, and Cheddar Man was a haplogroup I, why his Y-DNA was I as well. So this is, the genomic data is pretty consistent. Then you see the Anatolian hunter-gatherers in the Neolithic era. And you can see that even in the Anatolian hunter-gatherer population group, dark pigmentation is still dominant. And when we get into the Anatolian Neolithic farmers, this is when you start seeing the first pigmentation allele. Go on to early European farmers. This is where they mix with, it says the ANF migrants into Europe, mixes with the Western hunter-gatherers. Lighter pigmentation common, lactase absent, very rare. Then we move into the steppe pastoralist, the Yamanaya, which is another component as well. Let me see here. I want to see something, and I'll bring it on the screen if it's relevant. Ask ChatGPT something. What population group is the Yamanaya derived from? Okay, so I'm gonna put this on the screen. I think this is important to know, because out of the Cyto-Kino, two groups, it seems like, emerge. It says it's splitting here. Okay, all of it. Okay, but I'm gonna get it on the screen so the audience can see it. Yeah, man, we're gonna make this as subjective as possible, man. Make this airtight. So, Eastern hunter gatherers produce, or the Cytokino is from the Eastern hunter gatherers. It was another. I guess this is the fourth group now. Actually, the fifth. Right? You got the Western hunter gatherers. You got the Anatolian um, Western hunter gatherers, Anatolian farmers, um, Eastern hunter gatherers, Caucasus hunter gatherers here, and then you got the Proto Yamanaya ancestry here, um, which eventually, uh, is responsible for the Yamanaya, population later on. It gives this date right here. So, okay, they're saying it's coming from the Caucasus hunter gatherers. Okay. I also want to ask about Malta. How does Malta one play a part into European ancestry? Okay, it says Malta 1 is actually one of the key ancient genomes for understanding ancient North Eurasian ancestry, but its role in European ancestry is indirect and regionally variable, rather than being a primary ancestral population like the Villabrona or ANF. Here's how it fits into the framework you've been building. From Tagora to the ancient North Eurasian. So it contributed to the Eastern hunter gatherers indirectly. Okay. I think that speaks for itself. And I think that's where I'm gonna end the ancestry for contemporary Europeans. I think we've hammered that home pretty well. Oh, it wasn't even on the screen. Dang it. Put it on the screen. Go back through it one more time. So let me go back to the top and show y'all. Okay. So this is how the Yamanaya contributed into Europe and where it came from. You can see it stems from the Eastern hunter gatherers. Um, the Caucasus hunter gatherers split into its own group, but also contributes into Proto Yamanaya and then the Yamanaya into Europe. You can see that clearly. It says here that the Yamanaya approximately are 40 to 45 to 60% Eastern hunter gatherer and 40 to 55% Caucasus hunter gatherers. So, like I said, 5 components, right? You got the Western hunter gatherers, you got the Eastern hunter gatherers, you have the Anatolian farmers, you have the Caucasus hunter gatherers, and then you got the Yamanaya. So that's the 5 uh, main ancestral groups that contributed to contemporary Europeans. Um, and as I move further down and I ask that question about Malta, Malta is going to contribute gene flow into Eastern hunter gatherers. Where's that at? Past that? Yeah, here it is, right here. You see that chart right there. So me take a sneak peek at objectives. Measures, review results, and conclusions. Okay. I'm gonna finish doing this. Get this off the screen. This time, I'm gonna make sure I get this on the screen. There we go. Okay, so we went over the Yamaniya. And in the Bronze Age, got lighter pigmentation, widespread, lactase persistence increased strongly. And then by the Iron Age, is when that pigmentation and the lactase intolerance become very persistent. But we're going to see with the pigmentation, the depigmentation article that I'm going to share here. We're going to see that, you know, in their estimation, that depigmentation across Europe actually took place a lot more recent than even this AI generated flow chart. So yeah, I'm gonna get into that. I think that's next. Yes, it is. So hold on tight. We'll get the article on the screen. And I'm gonna line this up and make it make sense in the most non-subjective manner. All right, let me get this on the screen, y'all. I've actually read this on my short, or at least summarized it, man. But I'm gonna read the whole thing once again, just to bring it all together. Let me get it on the screen. All right. Now, I'm pretty sure this article came out in 2025. Let's go to the bottom and see what the print was. Yeah, 2025. It's titled The Evolving Pigment Palette of European Skin, Eyes, and Hair as Seen Through Ancient DNA. Let's read. And this is by Justin Jackson. Okay. University of Ferrara researchers in Italy have examined how European skin, eye, and hair pigmentation evolved over the past 45,000 years. This is even better because it's coming from Europeans that actually live in European. I'm sorry, not live in European, but live in Europe. Findings indicate that lighter pigmentation traits emerged gradually and non-linearly, with dark skin persisting in many populations well into the copper and iron ages. The study used the probabilistic genotype likelihood method to infer pigmentation traits from low coverage ancient DNA. Light pigmentation alleles became more common as Homo sapiens dispersed from Africa into regions with lower ultraviolet UV radiation. Dodge that hijack. That's a theory which is not supported by um, scientific data, empirical data. It's not supported. So once again, these are in my opinion, constructed narratives that are not supported by scientific data. But anyway, I'm not going to get too deep into that, but I just want to make mention of that. Evolutionary models suggest this was due to selective advantages related to vitamin D synthesis, as well as genetic drift and migration. However, the timing and pattern of pigmentation changes remain unclear. And I think part of the reason why it's unclear is because they keep, chasing this backwards, chasing these theories that are unproven. And I think that's part of the reason why the pattern doesn't make sense to them. And they haven't been able to figure it out. But anyway, in the study, inference of human pigmentation from ancient DNA by genotype likelihood, available as a preprint on BioRxiv, researchers tested the accuracy of pigmentation inference methods using two ancient genomes with high coverage. The 45,000-year-old Ishim individual from Russia and the 9,000-year-old SF12 individual from Sweden. A downstream experiment simulated low coverage conditions to compare three genotype calling methods: direct calling, imputation, and the probabilistic approach using genotype likelihoods. The probabilistic method was found to be the most reliable for samples with coverage below 8 times. This approach was then used to analyze a data set of 348 ancient genomes from Eurasia. Dark pigmentation was inferred for nearly all of the Paleolithic individuals, and they're ranging this from 45,000 years to 13,000 years ago, with only one exception showing an intermediate skin color. So they're saying they're not even intermediate. They're saying that these people are darky dark. I think it's very clear here. I'm gonna read this again. A downsampling experiment simulated low coverage conditions to compare three genotype calling methods: direct calling, imputation, and probabilistic approach using genotype likelihoods. The probabilistic method was found to be the most reliable for samples with coverage below 8 times. The approach was then used to analyze a data set of 348 ancient genomes from Eurasia. Dark pigmentation was inferred for nearly all the Paleolithic individuals, which ranged from 45,000 to 13,000 years ago, with only one exception showing an intermediate skin color. Wow. That pretty much matches up with the data that was previously shown in the flow chart. And I think this hammers it home even more than the flow chart does. But let's keep going. During the Mesolithic period, lighter eye colors became more frequent, with 11 out of 35 samples showing the light eye phenotype. These light eye traits were primarily found in samples from Northern and Western Europe, with dark hair and skin remaining dominant. Light skin tones first appeared in Mesolithic Sweden, but remained rare. And I love this article because it's a precise article. It's naming regions, it's naming countries, it's naming timelines. So, it's very difficult to, to misunderstand this particular article. They put it very plain and simple, for the reader. And it's no mistaking this. You know, it's putting these melanated people, and they're not just melanated. I shared the anthropological, or morphological features of these people as well. So they weren't just some melanated people. You know what I mean? They became more, uh, like contemporary Europeans over time, for sure. Just like I shared earlier. You know what I mean? But the Paleolithic period was just based on the anthropology, based on the genetics, in terms of the lineage. They were just some straight up Negroes. You know what I mean? People can feel how they want to feel about it. You know what I mean? The evidence speaks for themselves. And right around the time of Cheddar Man is when you start seeing that uh that shift into contemporary Europeans. But even still, you still had pockets of populations that still maintain their strict a strict Negro stock as well. And that's not necessarily pointed out in these studies that I've shared in this series. But hopefully I can grab some more evidence to speak to that particular part of it. But at least we can see that in the Paleolithic period, they were definitely Negroes, and their gene flow laid the foundation for later Europe. Even though the contributions into contemporary Europeans were more indirect down the line, and it wasn't, like a linear continuation into contemporary Europeans, you still get the gist of what I'm saying. So let's continue with this article. This spike in light eye pigmentation appears to be specific to the Mesolithic period. It suggests that for a brief interval in human prehistory, there was a higher occurrence of light eye trait compared to both earlier Paleolithic and later Neolithic Bronze Age periods. During the Neolithic period, pigmentation diversity increased, coinciding with the migration of Anatolian farmers into Europe. So once again, this is another genomic study that's building on everything that I've been saying. It's saying that pigmentation diversity increased, coinciding with the migration of Anatolian farmers into Europe. Lighter skin tones became more frequent, but dark phenotypes persisted, particularly in Southern and Eastern Europe. Hair and eye color also showed variability, with instances of red hair appearing in Turkey. All right, Copper and Bronze Ages, period being 7,000 to 3,000 years ago, saw continued increases in light pigmentation, but dark phenotypes remained widespread. By the Iron Age, which was between 3,000 and 1,700 years ago, light skin was nearly as frequent as dark skin, particularly in the northern and central Europe. However, dark pigmentation remained common in regions such as Italy, Spain, and Russia, just like Benjamin Franklin said. I mean, how many times do we got to repeat this? How many times? I'm gonna read that again. Copper and Bronze Ages between 7,000 and 3,000 years ago saw continued increases in light pigmentation, but dark phenotypes remain widespread. By the Iron Age, which was between 3,000 and 1,700 years ago, light skin was nearly as frequent as dark skin, particularly in northern and central Europe. So even in northern and central Europe, they're still not saying that light skin dominated. They just said it was just as frequent as dark skin, even in the northern European areas. So this whole nonsense about well, dark skin is not adapted for cold regions, and so you guys couldn't have inhabited those regions. You guys are not suited for those regions. From all of these studies that I put together, dark skinned melanated individuals, you might as well just call them Negroes, have been in these regions before the last glacial maximum, during the last glacial maximum, and after the last glacial maximum in these regions. And it goes on to say, however, dark pigmentation remained common in regions such as Italy, Spain, and Russia. Pigmentation changes appear to have been driven primarily by migration and gene flow rather than a linear pattern of selection. Didn't I just say that contemporary Europeans don't descend from a linear ancestry? That it was a lot of gene flow input, and that's pretty much what the article is about. You know what I mean? How much gene flow input in terms of the data that was captured, because they still indicated that there are still gene flow from populations that haven't been understood or captured. The spread of the Neolithic farming populations played a key role in shifting pigmentation traits across Europe. Genetic analysis highlighted key variants such as the SLC2-4A5 and the TYR, and others that contributed to these changes. The findings suggest that light skin became common in Europe much later than previously thought, and that pigmentation traits were shaped by a complex interplay of genetic and environmental factors over thousands of years. And that is the end of that article. I think that's pretty damning for the haters. I think so, man. Let's see what's up on next on the menu. So I've read the depigmentation article of aligned pigmentation with population divergence timing. A little self-explanatory in that article. I don't think there's any need for the PCA chart. How early was Y-DNA in the Levant? So for this, I'm going back to ChatGPT. I'm gonna get that on the screen right now. Let's get into it. We ain't gonna play games this evening. No. No, sir. We done playing with them in 2026. Y'all gonna have to go play with somebody else. And that is the real. Alright, so so for this is in the same thread. I gotta go back up earlier. I gotta find it. Shouldn't take me too long. And I think this is also going to spill over into here we go. The gene flow going into the Levant, into Africa, because I asked that question as well. And I'll probably just read the whole article on the Teferot, if I'm pronouncing that correctly. I'm going to go back all the way to when I started this part of it. Let's see. Might have gone a little bit too far here. Yeah, let me go back down. Let's see. I'll go back above this just to make sure. Make sure I ain't start something above this. Yeah. I'm glad I came back up here. Yeah. I was about to miss some. I think I started it right here. I asked ChatGPT, can you infer whether the Y DNA C in Eurasia was similar to Oceania in terms of genotype? It says yes, but only in a limited sense. We can make some inferences, but we cannot conclude they were genetically equivalent. There are three levels to distinguish: Y chromosome relationship. The Eurasian and Oceanian Y DNA C lineages were part of the same major haplogroup. You can see where it splits from CF to C and F, respectively. Ancient European is C1, and you can see here that it's marking the ancient Levantine as C. I'm gonna have to ask it about that because it seems like um, the more basal form of the C is actually in the Levant. Because even in our Aboriginal Australians, their C1B2. I'm gonna have to come back and ask that, man. So, genome-wide ancestry, it says there, this is where the answers changes. Ancient Europeans carrying Y DNA-C were not genome-wide identical to present-day Aboriginal Australians or Papuans. The available ancient genomes place Upper Paleolithic Europeans within early West Eurasian variation, whereas present-day Aboriginal Australians and Papuans form their own branch after tens of thousands of years of independent evolution, which is to be expected. However, because the split was relatively early, it's reasonable to infer that the earliest sea carrying populations were genetically more similar to one another than their modern descendants are today. However, we got to remember that, for example, around 50 to 45,000 years ago, sea carrying populations had only recently diverged. By 35 to 30,000 years ago in Europe, the Near East, Siberia, East Asia, and Sahel had already accumulated regional genetic differences. By the present day, those differences are much greater due to 40,000 years of independent evolution driven admixture, which is to be expected. Morphology, this is the area we discussed earlier. Some anthropologists have noted that certain Upper Paleolithic crania possesses features that they considered more generalized or that showed affinities with Australo-Melanesian comparative samples. Oh, so they made a direct comparison. Wow. At the same time, ancient DNA indicates that the Upper Paleolithic Europeans were already genetically differentiated from the ancestors of present day Aboriginal Australians. So I think this is where people get certain things mixed up. They think that because populations of Negro stock have changed genetics because of the land mass that they have derived on. Because again, a lot of the a lot of these genomic studies are, capturing, you know, fast um, mutating SNPs, and within those SNPs, they are capturing the mutations that are strictly based on, environmental pressures, things that transpire fast, which can make these populations appear more related to other populations that share the same landmass that don't have the same direct ancestral relationship. And so this is where the ambiguity comes in, and people say, "Well, see, these Australo-Melanesians, they over here in Southeast Asia or Oceania, and they got genotypes, consistent with that region of the world. So therefore, since you Negroes have said to have come out of Africa, they have nothing to do with you. That's the ambiguous argument that these people use. But there are two layers to this. You know what I'm saying? There are slow S&Ps that capture deeper ancestry, and there are fast S&Ps that are shared by many different population groups that are not related. You know what I mean? So when they generalize two populations that clearly don't share the same ancestry, they're not saying that they are closely related. They're saying in general they share uh you know this landmass in common, so to speak. So yeah, I felt the need to go ahead and nip that, man, because the Suedos, they like to take this and run with it, and basically over simplified, I think that's the correct word to use, and use it to their game. You know what I'm saying? But we're not doing that on this channel. Yeah, we smashing that on this channel. We getting rid of that on this channel for sure. But anyway, let's move on. At the same time, ancient DNA indicates that the Upper Paleolithic. We already went over that. So a cautious synthesis would be they may have retained some ancestral anatomical features that later became rare in Europe. They were not simply the same population as the ancestors of present day Australians. Like I said, that's to be expected because science doesn't work like that. If you've been on the landmass for thousands upon thousands upon thousands of years, you're gonna have some type of differentiation between two different populations or two different land masses. That's pretty simple to understand. So putting it together, if we imagine the situation around 50,000 years ago, it might look like this: Europe, Levant, Siberia, East Asia, Sahel, increasing regional differentiation over time. So yes, it is reasonable to infer that the earliest sea-bearing populations across Eurasia share more genome-wide ancestry with one another than their descendants do today. But it will go beyond the current evidence to infer that the Upper Paleolithic Europeans or Levantines were genetically equivalent to present-day Aboriginal Australians or Papuans. Ancient DNA instead supports an early shared paternal ancestry, followed by substantial regional differentiation across tens of thousands of years. This is how things get hypocritical, because they always want to separate, um, you know, uh, uh, different groups on different land masses when it's convenient, right? Saying that they obtain different different genomes along the way because they're on different land masses, right? So I know this is not a apples to apples comparison when we're talking about West Africans and African Americans, but just conceptually, they want to ignore this concept. When we're talking about lineages concerning the E1B1A, which African Americans carry and West Africans carry, and not allowing a differentiation between two population groups sharing the same haplogroup. Whereas here, we'll see that they'll use the same line of logic when they want to take away these Austro-Melanesian populations and separate them from Europe. I just want to point that out. One thing your question highlights is interesting research frontier. If we recover more genomes from 50 to 45,000 years ago in the Levant, Arabia, and South Asia and Southeast Asia, we'll be able to reconstruct much more precisely what those earliest sea-bearing populations look like genetically before they diversified into the regional populations documented later in the Upper Paleolithic. We cover more genomes. Yeah, that makes sense. I'm not gonna go over that part. That's another segue. Let's see what else I talk about here. Before I get to this, I want to talk about what I think I saw from our earlier discussion in this thread. It seems that most basal Y-DNA-C was actually found in the Levant. Am I understanding this correctly? Let's see what it says. Okay, it says not quite. There are two different ideas that are easy to conflate. The most basal branch in the Y-DNA phylogeny, the oldest ancient C lineage that has actually been sampled. Those are not necessarily the same thing. Well, that's pretty much what I just said though. I said as of what's been found. What we discussed earlier, we established that Upper Paleolithic Europe had Y-DNA-C. The Levant also had Upper Paleolithic Y-DNA-C. Y-DNA-C was distributed across Eurasia very early, but that does not mean the Levant contains the most basal C lineage. Under the current phylogeny, the earliest branching, most basal living branches of haplogroup C are generally found in East, Southeast Asia, and Oceania, depending on a particular subclade. For example, C1b lineages survive in parts of East Asia and Oceania, C1b2 and characteristic of Aboriginal Australians. Other early C branches occur in East Asia and South Asia. These living lineages are used to reconstruct the branching order of the haplogroup. Ancient Europe and the Levant. The ancient European C lineages and Levantine C lineages are extremely important because they show that C was once much more widespread than it is today. Okay, let's see what else is said here. However, they are not currently regarded as the most basal branches of haplogroup C. Instead, they represent branches of C that later disappeared from those regions. The simplified picture is early Asian lineages of then Levantine. So, I guess it's saying that the gene flow is going from north to south in Europe. So, I'm gonna nip this in the bud like this. So, what Pacific C haplogroup was found in the Levant, and what downstream structure did it have? Based on the ancient DNA currently available, the Pacific Upper Paleolithic Near Eastern lineages you are referring to is Y-DNA C1A2, formerly called CV20, or C6 in older nomenclature, or a lineage very close to it. One of the key pieces of evidence comes from the Anatolian hunter-gatherer genome, which carried Y haplogroup C1A2. Researchers note that this same lineage had already been identified among some of the earliest European hunter-gatherers, demonstrating that it was shared across Western Eurasia before the Neolithic. You can see here, it's even in Japan. Man, I'm cracking heads with this one. It's a wrap. I mean, it's never a wrap with the Swedos, man. They only want to view, listen to science when Negroes is just out of Africa. You know what I mean? When we talk about this type of information, man, they'll find a way to try to get out of it, but we're gonna make it hard for them. Early in the conversation, we were discussing the Levant, specifically the strongest published ancient C1A2 evidence is from Anatolia, which is adjacent to the Levant, but not the same region. The ancient Levant itself is less well sampled for this period, so it's more precise to say Anatolia, direct evidence for C1A2 in late Pliocene hunter gatherers. Europe, direct evidence for Upper Paleolithic C1A2. Levant evidence for closely related Near Eastern hunter gatherer populations, but the ancient Y chromosome record is currently sparser than for Anatolia. Okay, fair enough, man. Fair enough. So now I want to get into the Tafarat. That's an interesting name. I want to get into the Tafarat. We're going to get ready to wrap it. Man, that thing wasn't on the screen, man. Anyway, I'm gonna briefly show it. I'm gonna go back up there and briefly show it. Can't believe that. Yeah, so this is where it started. I asked ChatGPT, can you infer whether the Y-DNA-C is in Eurasia was similar to Oceania in terms of genotype? This is the answer. I'm just going to scroll on down. This is the part where it says the morphology is directly comparable to Australo-Melanesians, but the genetics are different, of course, because they derived on separate land masses. Like I said, that's to be expected. I'm going to go over this Y population thing right now. But I'm going to ask it another direct question before we get to this part right here. Matter of fact, we'll read this first and then I ask the question. So I only got to come all the way back up. Okay, so I say, okay, fair enough. Let's talk about the Natufians and the Terrafault specimen, how they align with this study. It says that's a great direction because the Natufians and the Terrafault individuals help bridge the gap between the Upper Paleolithic populations we've been discussing and later Holocene populations. Here's how they fit into the current picture. So it has Upper Paleolithic, it's got Near East and Europe, Fernald Vestinice and Near Eastern hunter-gatherers into the Villebrana, Epipaleolithic, and the Natufians descend from the Epipaleolithic peoples, whereas the Terrafault is in North Africa, and they are North African hunter-gatherers. and they have gene input. I guess the Terrafauna and the Tupians, they mixed together, and later on they produced the Anatolian Neolithic farmers. Interesting. So the Terrafauna individuals from Morocco are fascinating because the genome-wide studies indicate they were not simply sub-Saharan Africans nor simply Near Easterners. Instead, they appear to derive ancestry from at least two major components: a component related to late Pleistocene Near Eastern populations, often modeled as Natufian related or sharing ancestry with the same broader lineage, and a deeply African component that is not well represented by present day West Africans. Earlier papers often modeled the Terrafauna as roughly two-thirds Near Eastern related and one-third Sub-Saharan related, although later work has refined those models and emphasized that the African component itself is ancient and not well represented. Let me scroll this down. Represented by any single living population. That tells us North Africa was already genetically structured by 15,000 years ago. So I'm gonna bring the actual Terrafauna article up and try to find it quick. I'm gonna read some of it. That's not it. Here it is. Yep. All right. We'll go get this article, put this on the screen, so I can show that this is not just some AI making up some stuff. You'll see it directly from the article. Remove that. Stop sharing it there. And I'm gonna get this article on the screen. And make sure I share it on the screen. Yes, sir. Now we're cooking. Scientists analyze oldest human DNA from Africa. When did this come out? 2018. March 16, 2018. Researchers find connections from Moroccan Stone Age dwellers to ancient Near Eastern and Sub-Saharan African populations. An international team of researchers have sequenced DNA from individuals from Morocco during day. Now, I just want to point something out. Right? So, ChatGPT just indicated that this sub-Saharan type of genetics has not been seen subsequently in Africa, and the history of this signal is not well understood. So what I'm getting at is, I've said before that this sub-Saharan, genetic signal is a type, and it's, these types are not landlocked. They might be found saturated today in one particular landmass or region, in one particular lineage or two or what have you. But that does not, the evidence is not, it does not prove that the origin of the sub-Saharan signal is in Africa. And that ChatGPT just indirectly said this. And as a matter of fact, well, I'm gonna read this a little bit, and then I'm gonna go back and I'm gonna ask ChatGPT two things. I'm gonna ask it first, how do we know that the C haplogroup, well, actually, I already know, the Anatolians came into the Levant and put, their gene flow into the Levant, especially later on. But anyway, I'm gonna ask it anyway. So I want some more depth into it. Then I'm gonna ask it, do we know for sure that the sub-Saharan signal originated in Africa? Or what extent can we, how far can we, what can we determine, or how much can we infer from the sub-Saharan signal's origin? Yeah, that's what I'm gonna ask. So anyway, I'm gonna read this article. An international team of researchers have sequenced DNA from individuals from Morocco dating to approximately 15,000 years ago. This is the oldest nuclear DNA from Africa ever successfully analyzed. The study, published in Science, shows that the individuals dating to the late Stone Age had a genetic heritage that was in part similar to ancient Levantine Natufians and an uncharacterized sub-Saharan African lineage to which modern West Africans are genetically the closest. An international team of researchers led by Johannes Cross and Chongwon Zhong from the Max Planck Institute of Science Human History, Jena, Germany, and Epigenol Bazugar from the Institut National des Sciences de l'Archéologie et du Patrimoine, Rabat, Morocco, and including scientists from Mohammed V University of Rabat, the Natural History Museum in London, University of Oxford, Université Mohammed Premier in Guja, and the Max Planck Institute of Evolutionary Anthropology in Leipzig have sequenced DNA from individuals from Morocco dating to approximately 15,000 years ago, as published in Science. This is the oldest nuclear DNA from Africa ever successfully analyzed. The individuals dating to the late Stone Age had a genetic heritage that was in part similar to Near Eastern populations and in part related to sub-Saharan African populations. Notice here that they're saying that this is the oldest specimen. People try to conflate the findings of these hominins in Africa with, Homo sapiens. And they're telling you right now that the oldest specimen related to modern human beings in Africa is 15,000 years old. Whereas in Europe, you can see that they're finding specimens as old as 65,000. We're actually in the 6 digits old. I might even share that. I just started a thread today on that. I might share it now. I might keep that for later on another segment, depending on how time is permitting us. What else I want to read here? So it says the individuals dating to the late Stone Age had a genetic heritage that was in part similar to Near Eastern populations and in part related to sub-Saharan African populations. North Africa is an important area in history of evolution of our species. The geography of North Africa also makes it an interesting area for studying how humans expanded out of Africa. It is part of the African continent, but the Sahara Desert presents a substantial barrier to travel to and from southern regions. Similarly, it is part of the Mediterranean region, but in the past, the sea could have presented a barrier to interaction with others as well. A better understanding of this history of North Africa is critical to understanding the history of our species, explains co author said our Amzazi of Muhammad 5 University in Rabat, Morocco. In order to address this, the team looked at a burial site in Guat les Pigeons near, near Tafarat in Morocco, associated with the later Stone Age Ibermerism culture. The Morusians, the I, the Iberio-Morusians, the Iberio-Morusians. It sounds very similar to the Moors. Are believed to be the first in the area to produce finer stone tools known as microliths. Grata des Pigeons is a crucial site to the understanding of the human history of northwestern Africa, since modern humans frequently inhabited this cave intensively during prolonged periods throughout the Middle and later Stone Age, explains co-author Louise Humphrey of the Natural History Museum in London. Around 15,000 years ago, there is evidence for more intensive use of the site, and Ibero-Morusians started to bury their dead at the back of the cave. So the researchers analyzed DNA from 9 individuals from Terra Fault using advanced sequences and analytical methods. They were able to recover mitochondrial data from 7 of the individuals and genome-wide nuclear data from 5 of the individuals. Because of the age of the samples at approximately 15,000 years old and the poor preservation characteristic of the area, this is an unprecedented achievement. This is the first and oldest Pleistocene DNA of our species recovered in Africa, explains co-senior author Abigil, whatever his name is, due to the challenging conditions for DNA preservation. So I'm gonna ask ChatGPT, what was the mtDNA? What was the uniparental DNA for the Tafarot? Make sure I'm spelling that right. It's a tough one. It's a tough one. It can be keep it easy. Okay, so it was E1B1B. And mtDNA. Terra individuals carry several mtDNA haplogroups, primarily the U6A and the M1B. These two maternal lineages are strongly associated with North Africa today. Why this combination is interesting. The uniparental markers tell a somewhat different story than the autosomal DNA. Why DNA is E1B1B, the mtDNA is U6A and 1B. Autosomal DNA is a mix of Near Eastern related component and deeply African component not well represented by present day West Africans. This illustrates an important principle in ancient DNA. Y DNA traces one paternal line, mtDNA traces one maternal line. Autosomal DNA reflects the combined ancestry of all ancestors. These three do not always tell identical demographic stories. What time about that to tell y'all? How many times we got to tell y'all? So now I'm gonna ask you this question. So how can you infer indirectly that the Y DNA C haplogroup had input into early Levantine populations? This is a good example of the difference between direct evidence and indirect inference. At present, we cannot directly infer that Y-DNA-C made a major contribution to later Levantine populations solely because Y-DNA-C exists in the neighboring regions, but we can identify several observations that make it a plausible hypothesis to investigate. We know that ancient Anatolian hunter-gatherers carried Y-DNA C1A2. Upper Paleolithic Europeans also carried C1A2. These populations existed before the Neolithic. So there was clearly a Western Eurasian distribution of C1A2 during the late Pleistocene. If you map these populations, C1A2, and then to Levant, Europe is C1A2, to Anatolia C1A2, the Levant ties between or adjacent to regions where ancient C1A2 is documented. That makes it reasonable to ask whether similar lineages were also present in the Levant. However, a gap in sampling is not evidence that they were there. It simply means that we do not yet have enough ancient Y chromosomes from the relevant time and place. Autosomal evidence. The 2023 Nature paper infers that Near Eastern hunter-gatherer related ancestry contributed to the Villa cluster before the Neolithic. That demonstrates population interaction between Western Eurasia and the Near East. But autosomal gene flow does not tell us which Y haplogroup carried it. For example, the contributing males could have belonged to CE, CT-derived lineages now extinct, or another lineage not yet sampled. The autosomal data alone cannot distinguish among those possibilities. What happened later? By the Upper Paleolithic and Neolithic, the paternal lineages documented in Levant are mostly different, including lineages within E and other haplogroups. That tells us that even if C had been present earlier, later demographic events could have replaced or greatly reduced those paternal lines while leaving some autosomal ancestry behind, in which case we know for sure that it did. And so now I'm gonna go back up into the thread where it's talking about I'm gonna go back into the thread when it's talking about the gene flow going into the Levant from Europe. I don't know what I can ask. What haplogroup did the Near Eastern farmers carry? The earliest farmer in Anatolia were dominated by G2A, smaller numbers of C1A2 and H2, occasionally J. So we can see the population turn over here. The C haplogroup was more associated with the Anatolian hunter-gatherers, is what we see here. And so it seems like the Near East farmers were associated with Anatolian peoples, not necessarily the Levantine population. However, a lot of gene flow came from the Anatolians, and that's what I asked. Didn't the Anatolian farmers later contribute gene flow into the Levant? Yes, they did. In fact, gene flow between Anatolia and the Levant went in both directions over time rather than being a one-way process. Simplified chronology looks like this. Near Eastern hunter-gatherers split into Anatolian hunter-gatherers and Levantine hunter-gatherers. That's what I was getting at. And so the Natufians are direct descendants of the C haplogroup carriers. And these same Natufians were one of the earliest inhabitants of the Levant. These same Natufians contributed gene flow into the Terrafault, whatever the name is, in North Africa that had, the affinities with the Natufians and the Sub-Saharan Sigma. So I'm gonna ask it this. So what did the genome show? Ancient DNA studies indicate that Anatolian and Levantine farmers were closely related but distinct at the beginning of the Neolithic. Over the course of the Neolithic, there was admixture between them. Later Levantine populations acquired some ancestry related to Anatolian farmers, while Anatolian populations also received ancestry from neighboring regions. So yes, ANF related ancestry contributed to later Levantine populations, but it did not replace the earlier Levantine populations. Instead, the Levant retains substantial ancestry from local epipaleolithic and Neolithic populations, including Natufian-related ancestry, while also incorporating ancestry from Anatolia. A simplified ancestry diagram would be Natufians to the Levantine Neolithic to the ANF gene flow. This fits the broader pattern we've been discussing. One thing that has emerged repeatedly is in this conversation is that the prehistoric Eurasia was characterized by structured populations connected by intermittent gene flow. Rather than simple replacement, the evidence often looks like population A splits into B and C, and B and C admixed together. This is true for the Bulibrana and the Near Eastern hunter-gatherers, ANF and the Levantine farmers, the Western hunter-gatherers and the ANF Europe, Eastern hunter-gatherers, and the Caucasus hunter-gatherers forming the Yamnaya. Yeah, buddy. One clarification, when we say ANF contributed gene flow into the Levant, we are referring to autosomal ancestry. The paternal Y DNA picture is more complex. Although G2A was dominant in Y lineage among early Anatolian farmers, later Levantine populations retained and acquired a mixture of paternal lineages, including the E, J, T, and others. That means autosomal ancestry and Y chromosome history are related, but are not always identical. So your observation is correct. The Levant was not genetically static. It experienced multiple episodes of interaction with Anatolia before, during, and after the emergence of agriculture, making it a long-term crossroads between neighboring regions. Yes, sir. Now, this is the connecting question here. This is the last, this is the last thing I wanted to connect here. In terms of the Eurasian, make sure this is all on the screen. Yeah. Dang, man, I keep making one mistake, man. Oh, wow, man. So I'm gonna go back up. I'm gonna share this sequence. Okay, so I asked it what haplogroup did the Near Eastern Eastern farmers carry? It said G2A. Then I asked ChatGPT, didn't the Anatolian farmers later contribute gene flow into the Levant? It said, yes, they did. In fact, the gene flow between Anatolia and the Levant went in both directions over time rather than being a one-way process. Here's the flow chart is designed to represent that gene flow. It says that Near Eastern hunter-gatherers split into Anatolian hunter-gatherers and Levantine hunter-gatherers, which became the Natufians. And we know that the gene flow from the Natufians were found in the Levant and in northwest Africa as well, along with that sub-Saharan signal in the Tarfara specimen. The Anatolian hunter-gatherers later became the progenitors of the Anatolian Neolithic farmers. Dang, I still ain't got it. Okay, one more time. Let me get this right. I'm gonna get it right. So once again, I asked, what haplogroup did the Near Eastern farmers carry? It said G2A, more or less C1A2 and H2, and occasionally J. Okay. Then I asked, didn't the Anatolian farmers later contribute gene flow into the Levant? It says yes, they did. In fact, the gene flow between Anatolia and Levant went in both directions. Once again, Near Eastern hunter-gatherers split into Anatolian hunter-gatherers and Levantine hunter-gatherers. Both groups carried the C haplogroup. Like I said, the Natufians put gene flow into the Levant, and it spread that same gene flow into Northwest Africa, found in the terraform. Taphra. Ancient DNA studies indicate Anatolian and Levantine farmers were closely related but distinct at the beginning of the Neolithic. Over the course of the Neolithic, there were admixture between them. Later Levantine populations acquired some ancestry related to Anatolian farmers, while Anatolian populations also received ancestry from neighboring regions. So yes, ANF-related ancestry contributed to later Levantine populations, but it did not replace the earlier Levantine populations. Instead, the Levant retained substantial ancestry from local Upper Paleolithic and Neolithic populations, including Natufian-related ancestry, while also incorporating ancestry from Anatolia. The simplified ancestry diagram would be Natufians to the Levantine Neolithic and ANF gene flow. This fits the broader pattern we've been discussing. One thing that has emerged repeatedly in this conversation is that the prehistoric Eurasia was characterized by structured populations connected by intermittent gene flow rather than simple replacement. The evidence often looks like A is the progenitor of B and C, and both B and C admix. You see that there. This is true for the Villa Bruna and Near Eastern hunter-gatherers, ANF and Levantine farmers, Western hunter-gatherers and ANF in Europe, Eastern hunter-gatherers and the Caucasus hunter-gatherers forming the Yamnaya. So my next question is, in terms of the Eurasian autosomal signal that came into West Africa, which population, which Levantine or which Eurasian slash Levantine population group do you suspect input that gene flow? It says this is a really good question because it gets at the distinction between ancestral source populations and the population that actually carried the gene flow into West Africa. Based on the current ancient DNA evidence, we cannot identify a single named archaeological population with certainty, but we can rank the candidates. First candidate, late Neolithic, Bronze Age Levantine populations. Most population genetic studies that model the West Eurasian ancestry in many West African populations find that it is more similar to ancient Levantine and North African populations than to later Europeans. Those Levantine populations themselves were already admixed and typically carried ancestry from Natufian related groups, Anatolian Neolithic farmers related ancestry, and Caucasus Iran related ancestry. So the source was probably not pure Natufian, pure ANF, but a later Levantine population descended from both. Candidate 2, North African populations. Another strong possibility is that the immediate source was a North African population. For example, Natufians, Levantine Neolithic, North African populations, West Africa. This fits archaeological evidence for Holocene movements across the Sahara while it was much greener than today. Third, Anatolian farmer-related ancestry. ANF probably contributed indirectly. The sequence would be ANF to the Levant, to North Africa, to West Africa. In other words, ANF ancestry could be part of the ancestry that eventually reached West Africa without ANF themselves migrating directly there. Candidate 4, Natufian-related ancestry. Natufians are also an attractive candidate because they precede Levantine farmers. Later Levantine populations inherit substantial Natufian ancestry. Natufian-related ancestry has been detected in various later Near Eastern and North African populations. Again, though, I think the immediate source was probably later populations carrying Natufian ancestry, not the Natufians themselves. How this fits the papers you've been reading. One thing we've been building over the last several days is this chronology. Near Eastern hunter-gatherers, Natufians to the Levantine Neolithic, to the ANF admixture. Bronze Age Levant to North Africa to West Africa. Y'all pay attention to that gene flow, man. That model fits ancient DNA and archaeological evidence for the Holocene trans-Saharan interactions. Let's look at that tree again. Near Eastern hunter-gatherers who got their gene flow from Europe progenerate the Natufians, they progenerate the Levantine Neolithics, they progenerate ANF admixture, they progenerate Bronze Age Levant, they progenerate North Africa, and they progenerate West Africa. So the father, the granddaddy of this whole tree is Astralo-Melanesians. And remember, they made a direct morphological connection between this population of Europe or Eurasia carrying the C haplogroup to Austro-Melanesians, and there is extremely high affinity in the morphology. I'm just saying. Okay, so my last question would be, what was I going to ask? So the Natufians could have been capable of contributing the Neanderthal and Denisovan variant that have been observed recently in West Africa. Yes, they could have, but with an important distinction between carrying archaic ancestry and being the original source of that ancestry. Here's why. First, Where did the archaic ancestry come from? By the time the Natufians appear, Neanderthal interaction had already occurred tens of thousands of years earlier, probably shortly after modern humans dispersed into Eurasia. Gosh, that hijack. Modern humans leave Africa. Neanderthal, we understand that. This means the Natufians inherited Neanderthal derived DNA. They were not the population in which that admixture first occurred. Could they transmit into Africa? Yes. If an Natufian related or later Levantine derived population contributed ancestry to North Africa and then West Africa, they would also transmit whatever Neanderthal derived alleles they carry. The same logic applies to the two to Denisovan derived alleles, although the picture is more complicated. Neanderthal versus Denisovans. There is an important difference. Neanderthal ancestry is present in essentially in all non-African populations, therefore could plausibly enter Africa through any West Eurasian back migration. Denisovan ancestry is much more unevenly distributed. Present-day West Eurasians generally carry very little Denisovan ancestry compared with populations in East Asia, Oceania, and parts of South Asia. Therefore, if Denisovan derived variants are found in West Africa, they do not automatically imply Natufian ancestry. Those variants could reflect ancient shared variation, multiple episodes of back migration, indirect transfer through populations that already carried low levels of Denisovan ancestry, or for Pacific alleles, independent evolutionary histories. Researchers evaluate each variant individuals rather than assuming a single source. Relating to your earlier chart, if the Eurasian component entered the West Africa through a Levantine North African route, the sequence could be Neanderthal admixture with Near Eastern hunter-gatherers, the Tufians, later Levantine populations, North Africa, West Africa. One interesting point. This ties into something we've discussed before regarding the Levantine-related Eurasian signal detected in some West African and African American genomes. If that ancestry derives from late Pleistocene or Holocene near Eastern populations, then it would naturally be expected to carry some Neanderthal ancestry because all sample late Pleistocene West Eurasian populations do. The question that remains active in research is which Near Eastern or North African population were the principal contributors and when those gene flow events occurred. So I think the strongest conclusion supported by current evidence is yes, the Tufian-related populations were capable of transmitting Neanderthal-derived variants because they already carried them. For the Denisovan-derived variants, that situation is less straightforward. Some Denisovan-associated alleles could have been transmitted through West Eurasian populations, but if West African populations carried Denisovan-derived variants, each case needs to be evaluated individually because West Eurasians generally have much lower Denisovan ancestry than populations further east. So I'm going to ask this question. Wouldn't the plausibility come or likely through Y DNA C carriers being that they first derived, or I say first differentiated in a region with a significant Denisovan presence. The reason it goes like this: under the current phylogenetic model, haplogroup C diversified early in Eastern Eurasia, often modeled in South or Southeast Asia. Those regions are also where interactions with Denisovan-related populations are thought to have occurred. Therefore, early sea-bearing populations could have had opportunities to acquire Denisovan uh, ancestry before dispersion across Eurasia. That chain of reasoning is plausible. However, there are two important caveats. Y DNA is not a proxy for autosomal ancestry. A Y chromosome tracks only one paternal lineage. Imagine an early sea-bearing population, Y equals C autosomal, 2% is Denisovan. Several thousands of years later, after repeated admixture, Y chromosome can persist, while most of the autosomal genome changes through an admixture and genetic drift. Conversely, population C, Y equals G2A, autosomal inherited Denisovan derived variants. Population could lose Y-DNA C entirely, but retain some Denisovan derived autosomal alleles inherited through earlier admixture. Western Eurasian sea carriers, the Upper Paleolithic C1A2 carriers in Europe and Anatolia were already part of West Eurasian populations. Genome-wide analysis of those individuals do not show the elevated Denisovan ancestry seen in present-day Papuans or many East Asian populations. Instead, they fit within the range expected for early West Eurasians who generally have substantial Neanderthal ancestry and relatively low Denisovan ancestry. So while those seed-bearing populations could have inherited some Denisovan-derived alleles from much earlier ancestors, the currently sequenced genomes do not indicate that they retain unusually high Denisovan ancestry. So could an earlier seed-bearing population have been different? Yes, that's an open possibility. One could imagine a scenario like early sea populations interact with Denisovans, splitting of West Eurasian and East Eurasian branches, much later admixture, higher Denisovan retention, and for the West Eurasians, they reduced Denisovan signals. This is a biologically reasonable demographic model. The question is whether the ancient genome supported. At present, we have strong evidence that Denisovan ancestry is highest in populations of Oceania, parts of East and Southeast Asia. Ancient West Eurasian genomes, including those carrying C1A2, have not shown comparable high Denisovan ancestry. Relating this to West Africa, if Eurasian ancestry in West Africa came from late Pleistocene or Holocene Levantine North African populations, those source populations almost certainly carried Neanderthal-derived DNA. Whether they carried enough Denisovan-derived ancestry to explain Pacific Denisovan-associated variants of West Africa depends on which variants are being discussed. Some variants are widespread across non-Africans and at low frequency, while others are much more concentrated in Eastern Eurasia. Researchers therefore evaluate individual loci rather than assuming that they all came from the same source population. So I think your hypothesis identifies a legitimate avenue for investigation. Early seed-bearing populations may have served as one route by which the Denisovan-derived alleles spread across parts of Eurasia. The current evidence, however, does not allow us to conclude that the C1A2 populations of Upper Paleolithic Europe or Anatolia were the principal source of Denisovan-derived variation later observed in West Africa. That would require either direct ancient genomes showing high Denisovan ancestry in those Western seed-bearing populations, or autosomal analysis linking the Pacific Denisovan-derived variants in West Africa to those populations. Last question. So, what would be the most likely mode of transportation of the Denisovan variant. Says, given what we know today, the most likely transportation of Denisovan derived variants into West Africa would have been via autosomal gene flow through populations that already carried those variants rather than by any particular Y chromosome haplogroup. Here's how I rank the scenarios based on current evidence. Holocene, Near Eastern North African back migration, best supported. This is the scenario most population geneticists currently favor for the West Eurasian ancestry observed in many West African populations. Early Eurasians, admixing with Neanderthals plus low Denisovan ancestry into Near Eastern, they derive into Near Eastern populations, from there to North African populations, into West Africa. If those Near Eastern North African populations carry some Denisovan-derived alleles, even at low frequency, they could have transmitted them into West Africa. It says too, late Pleistocene back migration, an older movement from Western Eurasia or northeastern Africa into Africa before the Holocene could also have introduced archaic-derived variants. This remains an active area of research because the ancient DNA record from North Africa between roughly 50 to 20,000 years ago is still sparse. Early seed-bearing populations. This is the hypothesis you've been developing. The logic is early seed-bearing population, possible Denisovan admixture, splits in the West Eurasia and East Eurasia, the West Eurasia branch into Near Eastern populations, into North Africa, and then into West Africa. This is biologically plausible if those seed-bearing Western Eurasian populations retain Denisovan-derived variants. The limitation is that the ancient genomes we currently have from Western Eurasian C1A2 carriers do not show the elevated Denisovan ancestry seen in populations farther east. That doesn't rule out the hypothesis, but it means it's currently demonstrated. Then independent archaic integration within Africa. Another possibility discussed in the literature is that some archaic-derived variants in Africa populations may come from archaic African hominids, not Denisovans themselves. Definitely plausible. And so I've already thought that, some of these variants, whether they be a Neanderthal or whether they be Denisovans, could actually be conflated with archaic ancestry within these Negroid populations, especially observed in West Africa. They might not necessarily be Neanderthals or Denisovans. I'm open to that, and I understand the possibility of that. But nonetheless, this gene flow is coming from seed bearers. And I'm gonna land that home. Even in the first scenario, C haplogroup carriers are still a part of that gene flow, correct? Yes, they could be, but we have to distinguish between ancestral contribution and direct persistence of the Y chromosome. If we follow the current mainstream model, the sequence could look something like this. Early seed-bearing populations in the Near Eastern hunter-gatherers, Tufians, Levantine Neolithic. And Nephad mixture, later Levantine populations, North Africa to West Africa. In that framework, yes, descendants of early sea-bearing populations could contribute autosomal ancestry to later Levantine populations, even if the CY chromosome itself became a rare or or disappeared through later demographic events. So I think I'm gonna leave it here. I don't think it's gonna be a need for a part three. I'll think about it. I might think about some other stuff, and I might want to come back. I'm, want to add to it, But I think this has been decent enough. It's a little choppy because I kept forgetting to get certain prompts on the screen, but I think it'll do. You know, with part one um, and this part put together, I think I conveyed the message that I was trying to convey. You know what I'm saying? So I'm gonna leave it right here and wrap it. And I'll probably segue into other segments based off of this. But for right now, that's it, man.